From Institut Fresnel in Marseille in France, and I'm going to talk to you about polarized microscopy in fluorescence and super resolution imaging. I'm coming from a team who is developing new microscopy tools for bioimaging in general. I'm going to give this presentation in English, but you can of course ask your questions in French at the end of this talk. So our main limitation in optical microscopy is, as you know, the resolution of what can be imaged. And on the left here is an example of observation of actin organizations in a cell. And as you can see here, the background between these very highly linear fibers, stress fibers, is quite blurry. And the reason for this is that there is actin there, but the organization of it cannot really be imaged with optical microscopy in its classical way, either confocal or spinning disk or wide field or whatever you want. The reason for this is that the smallest size and smallest organization you can image in optics is about hundreds of nanometers due to optical diffraction limits. This is not something limiting in electron microscopy, where we can see very beautiful and precise organizations of actin filaments there. However, what we want to do is to image this kind of organization, but in live cells, for instance, in very high dynamic rate where cells treatments are not required. So actin organization is actually made of orientations of actin filaments. And this is this top figure that I'm showing here, where we can see that depending on the region of the cell you're looking at, these actin filaments can take very different type of orientation organization, from very highly wide aperture angular networks, such as found, for instance, in the lamellipodia of cells, to very highly parallel organizations, which can be found, for instance, in stress fibers. These different types of organization are differentiated and discriminated by clear orientational properties, and this is what we're going to try to measure here. This is not only about structure, and it can be about properties of single proteins, such as integrin here in this lower part of the graphs. Integrin is known for its property in recognition of a substrate, for instance, binding to an external part of the cell. There are lots of studies done in tracking, for instance, single integrins, being able to see its different behaviors between highly diffusive translational behavior to stopped one when they reach focal adhesions. But what is known as well is that integrin changes an orientation in its external part of the cell domain upon activation. And this is something that cannot be studied by pure localization microscopy. So here again, orientation is important. And this is something that we would like to probe with pure optical tools. And this is where polarization will come into play. So what do we mean by actually organization? It is orientation. However, this imposes that we rely on fluorophores to report our proteins organization and proteins orientation. And the requirement for this is that a single fluorophore needs to be attached to our protein of interest in a quite rigid way. So this linker has to be as rigid as possible. And this is true as well when studying membranes, for instance, where our lipid protein needs to be highly oriented in our membrane. So this is overall a requirement, and as we can imagine, this is not something which is very traditional, because usually we try to attach our proteins to labels in the least intrusive way, which means that the label is quite far away with a high mobility possible. So this is something to adapt, of course, but overall what we are going to measure is the orientation of this single fluorophores here, or ensembles of fluorophores, as a reporter of our proteins organization. And this can be from very highly ordered one to completely isotropic. This variation of orientational behaviors is going to be called orientational order. This is what we want to probe in the end in microscopy. And this is about looking at either ensemble of molecules, such as here, what would be represented in the focal spot of a microscope, or a single molecule which would evolve in time. This angle that you see here is in the end the angular aperture explored by these molecules, either ensemble or time-dependent single molecules, and this is what we want to measure. So to do this, we will use light polarization. This is a property of light which makes it directional. It tells us how the electric field of light, which is oscillating rotating in time, is going to be oriented, and this can be either linear, upon different directions, or elliptical, or even circular. This is something we can control in optics. And this polarization is going to act as either an excitation control or a probing control in our fluorescence properties. So how do we actually play with polarization of light as an excitation control? And this is by tuning absorption. As you may know, polarization of light is actually acting in a fluorophore by helping bound electrons to oscillate within the body of this molecule. And when the polarization of light is going to be parallel to this conjugated direction, this will enhance absorption to a high level, while when it's orthogonal, there is very little absorption going on. This is highly related actually to the way that electrons are behaving in the molecular body, and what is of interest for us is this absorption dipole, which is in fact a transition dipole, which is the way that different orbitals in these molecules are going to be coupled through the dipole operator. So basically the structure of the molecule is driving the direction of this absorption dipole, which is itself reacting to the polarization of light. On the other hand, fluorescence is not only absorption, it's also emission of light. And this is another step of this process, which makes emission highly polarized as well. This is a bit similar to having an emission dipole, which would be parallel to its molecular body. And if I rotate the polarizer here in my emission path, I'm going to have a variation of the emission of light, which will be high when my polarizer is along the molecular body and very low when the polarizer will be perpendicular to it. All this is related to the fact that when fluorescence is operating, when a photon is emitting, this comes from something similar to an emission dipole, which is a radiating light and which will be collected by a microscope objective. So overall, there are two ways to measure molecular orientations, either playing on the linear dichroism, which is a way to tune absorption, or playing on the emission path with a polarized detection. If you play on both of them, then you're even able to measure the way that molecules are rotating in real time. And this is called typically fluorescence anisotropy, which is a very old technique used in cuvettes, for instance. So here what we want to see is not necessarily reorientation, but it's organization. So we're going to play either on the absorption or on the emission steps. So let's go for the absorption first. The way to do it is actually to rotate a polarization in the sample plane. So let's take, for instance, a single molecule, which is going to be excited by a rotating polarization. This will lead to an oscillation of the fluorescence signal, which is maximal when the polarization is along the dipole direction. This allows to point its direction, and which is highly contrasted, as I said before, because no absorption is occurring when the light is perpendicularly polarized to the direction of the dipole. However, in general, we look at many molecules, or we look at a wobbling single molecule. In this case, many different of these curves are going to be added up, which are shifted in angle, and it is contained typically in a cone distribution, as I represent here, which makes this modulation of fluorescence losing contrast. So the loss of contrast here of this curve is highly related to the organization of these many molecules. When they are exploring many different orientation angles, then I will tend to have a flatter modulation of fluorescence. And this is signature for this organization molecular order angle. However, the maximum can still be pointed, and this gives me the orientation, the mean orientation of where these many molecules are pointing. So in practice, the way to do it is to build a setup which is any kind of fluorescence imaging you can think of. However, what we will do is we will tune the incident polarization angle, which should be an alpha here, by whatever tool you can find to rotate the polarization. This can be the rotating half-wave plate, this can be a Pocket cell, and to record then afterwards per pixel a modulation, which we treat by extracting two important parameters here: the mean orientation of this population of molecules measured in this precise pixel location, and its molecular average, angular aperture, which is psi here, which will be the molecular order. So physically what it corresponds to is per pixel to a measurement of all these different molecules organized together. And this is an example of a typical biological sample that we're looking at. So look at actin. Here is a fixed cell where we have bound Alexa 488 to actin filaments via phalloidin conjugates. And what will happen is we allow, of course, these molecules to have a certain mobility. This is not precluded by fixation. There is still water in the sample. But what we look at is the way that these filaments are organized locally per pixel, which is itself a given organization, which we call psi here. So psi is due to the way that filaments are going to be oriented within this focal spot and the way that molecules are going to wobble in time. So what we measure in the end is a fluorescence image, but per pixel we add the information of the modulation of the fluorescence signal, which is due to the rotation of the excitation polarization. And we encode that in a color, which is called molecular order, from very reddish, if molecules are exploring a very isotropic orientation of angles, to very bluish, which means that molecules are highly organized. And again, remember that here I have both contributions contained in these ensemble measurements, the orientation of these actin filaments, but also the way that molecules are able to wobble. So here, for instance, typically this yellowish color you see in these actin stress fibers is quite a high angle, and we will see later that there is a high contribution of the fluorescence molecular wobbling here in these values. On the top of the molecular order, we measure orientation, as I told you. And this is showing here that these Alexa phalloidin conjugates are following the direction of the actin filaments in the sample. The way we represent both information, and this is work that we have developed in close collaboration with Manos Mavrakis, and this is a PhD work of Caio Rimoli, is to encode each of these pixels as a stick, which orientation is going to be the raw orientation, the mean orientation of these molecules, and the color of this stick will be the molecular order, from very highly organized in blue to very highly disorganized in red. So these are examples of what we have achieved with this method, and this is one of the most representative one, where we have been able here to detect the fact that actin is highly organized in certain parts of the cellularization of the Drosophila embryo development, and that this is highly related to another cytoskeleton protein that is present with actin at the basal part of the cellularization process. So here where we see septin by another fluorescence marker, we also see that the organization of actin is becoming more narrower than what we see at the apical part of the cell where septin is not present. And looking at this view from the top, indeed, all these sticks are very highly disorganized on the apical plane, while they are highly organized around the contour of these cells in the basal plane. And this is highly related to some structures that have been observed in parallel in vitro, where septin has been shown to be able to provoke some kind of aligned but curved orientations of the actin filaments here. Another example that I wanted to show you is in cell membranes. It's not only limited to actin, of course, but the way to observe membrane proteins has to be, of course, developed in conjugation with the way that the dyes is going to be attached to the membrane domain that we want to observe. So this is an example of what people have been able to do in CIML in Marseille, attaching a GFP to a protein part, which is intracellular, and this was to study TCR, which is a T-cell receptor. And these people have shown that if you attach actually a GFP only by one single arm here, there is a high disorganization of the protein at the membrane of the cell because of its floppiness. It's not able to report per se the orientation of a given protein in the cell membrane, while attaching it by its both N and C terminals is able to immobilize somehow this protein. And this can become therefore a reporter of what will occur later when the TCR receptor part will be changing directions, for instance. So this was about average measurements about many proteins, many dyes in a given focal volume of our microscope. However, there are many interpretations of molecular order at this stage. Many means that this can be a disorder at the spatial level, where different molecules can be static but exploring many different directions, and this is what we would call organization. But as I mentioned on actin earlier, this can as well be a disorganization at the time level, which is floppiness of the dye. And these two levels cannot be disentangled in these ensemble measurements. This is something that, of course, can be circumvented by looking at relative measurements. However, defining per se a structure which will resemble electron microscopy is not entirely possible at this stage if we do not know how much wobbling angle is explored by the single molecules. So a way somehow to observe a structure, such as people would do in electron microscopy, is to go down to single molecules to disentangle the wobbling behavior from the average direction behavior. And the other advantage of single molecule detection is, of course, that you can reconstruct a super resolution image. And this is an example for stone, where each single molecule would be also reporting a localization, helping to reconstruct a given filamentous structure, as for instance here in microtubules. This is, of course, depending on the number of photons that you detect, and the higher number of photons leads to a higher localization precision in this reconstruction. The only drawback of looking at single molecules, of course, is that you cannot, especially for super resolution imaging, you cannot detect for too long a single molecule. So this is why we privileged here to measure disorientations from single molecules, a detection, emission, polarized detection. So in a way, the idea here is to not anymore photo select any absorption orientation. So we excite here our molecules with circular polarization, but we're going now to play with the detection path to introduce, for instance, polarizers. So in a way, it's working very similarly to what I showed to you before in the absorption tuning. Here, what we will do is we will rotate a polarizer at the detection path of the microscope, and we will detect a modulation, which is now due to the orientation of this emission dipole. So the maximum here signal is shown by the direction of this dipole in its plane, while the contrast is showing if the dipole is alone or if it's wobbling. So here, a way to measure that in a single shot is actually to separate polarization channels in different polarization directions. So what we'll do is we will use 4 different angles of polarization to measure the contrast of this curve and its mean direction. So in a way, to get similar parameters as before, except that here the mean angle is the mean angle over time of a single molecule, and the delta is the C angle, which was the molecular order before, for a single molecule. So it is somehow its wobbling explored angle. The points to be aware of here is that what we are looking at is a projection of the information in a polarized way, which is forcing the signal to be in the plane of the sample. This is the plane where we manipulate polarizations. So what we have is a 2D picture. It's a projection of a possibly 3D orientation in the plane of the sample. And as you can see here in this little sketch, if the cone of wobbling of a single molecule is highly tilted off plane, then its information will be highly biased, because it will look like if it's a very isotropic organization. So we have to be very aware of that. So actually something very specific happens when you tilt a single molecule out of plane, and this is what we need to play with somehow to have this information. When the dipole is lying in plane, if you look at the back aperture, the back focal plane of the microscope objective, you're going to see a pattern of light which is very connected to the way that the dipole has been emitting light, and this is looking like a donut-like emission such as this one. So at this back focal plane here, I'm looking at a repartition, a pattern of light which is connected to this radiation pattern. It's also locally polarized along the direction of the dipole. When the dipole is tilted off plane though, then the pattern is going to lose its symmetry and to become a bit more tilted towards one of these edges here, with some polarization which become more radially oriented. And it's more and more radial-like when the dipole is going to be tilted completely out of plane. So when the dipole is along the propagation direction, then the emission energy is located very at the border in a homogeneous way, and the polarization is radial. So this is a signature that people have used a lot to observe 3-dimensional orientations of single molecules. Overall, what we want is a very high numbers of parameters. We want to measure a position of a single molecule. We want to measure its orientation in 3D, which means 2 average angles, in plane and out of plane, plus a wobbling angle, which is a very hard parameter to measure, actually. So a lot of different strategies have been developed. Some of them consist in putting in this backfold plane where the pattern of the light emission is the most marked, to place a faceplate, which is going to transform the shape of your PSF, which was generally Gaussian-like, into a shape which consists of different spots. These orientations of spots or their relative emission intensity will be a signature of the dipole orientation. This method, however, is losing most of the time either one or the other parameters among position or orientation. Other tools are defocused imaging, which is actually a way to visually measure the way that the dipole is having its radiation pattern. And another approach is actually to project the emission on different orientation polarization angles. However, this projects, again, this information in 2D. Among all these directions, there is no ideal method yet developed to measure all parameters at once in a very easy way. So what we have started developing is to keep the direction of looking at the 2D polarization response, which is a polarized one in the sample plane. The reason for that is that most of the samples that people are looking at in measuring organization can be perfectly flat samples, and this is what we were focused on to start with. The only thing to be careful about is that to be able to measure the most accurately possible polarization angle or an orientation property in 2D, it is necessary to remove the part of the emission that is related, that is due to this off-plane orientation. And the way to do that is to reduce a bit the numerical aperture. So to reduce a bit the numerical aperture means that we can put in the detection plane a reduced diaphragm that is going to look at only the small emission angle in our setup. As I said before, to measure in-plane polarization angle plus the wobbling information, 4 angles of detection polarization are needed. So this is why we developed a setup, which is going to appear in archive very soon in the coming weeks, which is based on the detection of 4 different channels. As you can see here, these are single molecules blinking, but a single molecule is present in 4 channels, which are polarized in 4 different polarization directions, which allow to explore overall this whole modulation curve that I was showing to you before. The way to do it is to split the beam into 2 different paths, which are themselves split into 2 different polarizations, which explore these 4 different directions overall. There are very simple analytical ways to extract the information that in the end we are looking for, the mean orientation of these single molecules and their wobbling angle. So this is the work of the PhD of Valentina Curcio. We have, of course, developed a way to locate these molecules in 2D. This is also transferable to 3D localization methods. So this is an example of what is observed, and this is the most simple actin sample we can think of. It's a single filament of actin with which attached are alexafluor 488 molecules with phalloidine. So this is this linker that you can see here, which is a very small part which binds into the groove of the G-actin domains. What we have observed here is that these molecules, so here one stick is actually one single molecule, which color is the wobbling of this molecule and which direction is its in-plane direction. What we have observed is that these molecules follow well the orientation of the F-actin, of the single actin filament, and that they can be overall characterized by a wobbling angle which is quite high, and this is about 90 hundredths degrees, as you can see here, while their mean direction follows quite well the direction of the filament. So this is actually understood by the fact that this dye is not exploring a single frozen direction. In time, it is able to wobble a bit, to change a bit its directionality by exploring different ability of direction, as was modeled by our collaborators in Toulouse. Now, if we move to a more complex sample, which is a cell, we have been able to observe that overall, on average, the mobility of the single molecules were quite close to this value that I was mentioning before, 100 degrees. And its orientation, of course, is following the actin filaments, which is something that we have already observed at the ensemble level. But as you can see and compare from the angles that we were seeing before from the molecular order, there is a large contribution of the average molecular order from an ensemble that is due to the wobbling of these single molecules. And this was a very important result that we got here from single molecule data. Looking a bit closer, actually, we can observe that single molecules follow still well stress fibers, which means that the packing of these stress fibers is actually, as people have reported in electron microscopy, very highly parallel. However, in some other fibers, such as the dorsal one here, this corresponds to this square, we can see roughly quite higher disorder of these little sticks. They explore very quite broader range of angles, and the color of these sticks is becoming more red, which means that there is an apparent higher wobbling. The reason for this is actually, as we understand it from analysis, is that these little molecules here that you see as a highly wobbled ones are actually biased wobbling measurements from highly tilted molecules. As I said before, we do reduce the numerical aperture to provide a selection of molecules that are lying in plane. However, it's not a 0 to 1 filtering, so we can still observe some degree of plane orientation molecules. And if you look at this bias graph here, we can see that even for a wobbling value of about 90 degrees, if I tilt a filament of plane by 30 degrees here, I'm going to have a bias on this delta measurement of about 40 degrees. So it means that the delta measured is not 90, but 90 plus 40 degrees, which is a higher value from what it should be if the molecule was in plane. So a way to actually only look at molecules that are in plane is by providing a further filter out of these molecules, looking, for instance, at delta values that are only small ones, lower than 100 degrees. And doing that, the sticks, the orientations of these single molecules become more parallel to the observed fiber. So actually, dorsal fibers, as observed here, could be visualized as a bundle of filaments that are highly parallel, except for a population of them which is tilted off plane and exploring highly more various directions. This is also something we have observed in focal adhesions, which are structures which tend to get off plane because of this attachment to fibers that are going to lie later to tilt later off the plane of the sample. So we have extended this study to other parts of cells, and this is an example of B16 cells which have been given by Clemens Röttner in Braunschweig, studying specifically the lamellipodia part of the cell, which is this front of migrating cells, which is very important for cell mechanics. All this part here is actually a bit reddish, so as you can see here from the delta value, so it means that there are populations of filaments that are also tilted off plane these structures, which was already quite interesting to see, as in contrast to the stress fibers that lie in the plane. And looking a bit closer to the way that these filaments were oriented, we can see first in the STOM image that it's very hard to disentangle any kind of organization in this filament meshwork because of their high density. This is basically the high limitation of STOM here. However, in terms of orientation, we can clearly visualize in these regions here structures which is not completely isotropic. And this is an example of histogram where we can observe a bimodal distribution of actin orientations, which resemble somehow what people have observed in electron microscopy, most probably due to the ARP2-3 crosslinker, which forces actin to be branched here. This could be observed at very different scales here in STOM, and it is clearly highly different from what we can see from stress fibers, which are clearly single directional structures, as you can see here in this other part of the cell. So the last part that I wanted to address here is actually the most challenging one, is how to actually measure three-dimensional orientation plus three-dimensional position of single molecules. And as I mentioned before, there are ways to do. And the way to do it is to filter by a Fourier plane, smart filtering, the way that the image of a single molecule is going to occur. And we have actually exploited a method that has been inspired by development from Miguel Alonso and Tom Brown in Rochester University, which was the use of faceplate, which is not only encoding phase, but also orientation of polarization. So this is like having little locally birefringent faceplates that are spatially patterned. And these people have shown that by measuring the state of the polarization of light going through such a faceplate, it is possible to translate it into measurement of both the polarization state of the light, but also its depolarization state. And this is giving shapes that are looking like little donuts or croissants, such as this one. The modeling actually that we have looked at with Miguel Alonso is to look at the way that a single molecule was actually emitting through a microscope, projecting after passing such a faceplate, projecting two different left and right circular polarization of the light, of the emitted light. And this can be modeled by field propagation through this full microscope and phase birefringent faceplate. So what is interesting here is that these croissants are actually rotating with the counter-rotating with the z position of the dipole, which is giving us a readout of the z position of the dipole, plus the possibility to observe its off-plane orientation by a big change of the shape of this PSF. So this is a very important part here, because a single shape of PSF projected into these two different directions is able to give you an unambiguous determination of the 6 parameters that we're talking about: orientation, position, plus wobbling, because when you start having some mobility of angle in this molecule, you kind of blurry this PSF shape. So all this is sortable by a projection of some basis. So I'm not going to detail the whole part here, but you can look at this recent paper about that. And experimentally, it has been possible to reproduce such PSF, which are actually not as expanded as what we have seen in previous papers in PSF engineering. They are quite still small, only twice the size of the single PSF. We have actually modeled as well the precision that you can get either on the spatial coordinates or angular one, and they are very close to what one can have in traditional single molecule localization methods, but also degrees like orientations, precisions, plus maybe 10 degrees precisions on the mobility of a single molecule on average, which is quite precise and quite uncorrelated from each other, which is also an interesting part here of these methods. And globally, we have started measuring using this method on single filaments to observe that single molecules could be encoded both in position, in angle, 3-dimensional orientation, in wobbling, and in pseudo-dimensional position as well. So to conclude, I have shown you that polarization fluorescence is actually able to give you more information than pure fluorescence image here, either in Ensemble, where we can compare different kinds of organization based on the way that polarization responds to absorption of molecules. And in single molecule-like measurement, it's even more powerful, because you can start looking at organizations which are right now only observable by electron microscopy. So there are actually still lots of developments to do in terms of making these methods optimal. Optimal means non-ambiguous, robust to a very low number of photons, insensitive to proximity of a surface, for instance. And this is there is a nice paper actually that has been published in this direction recently. I would like to thank, to finalize, my colleagues, the biologists, especially Manos Mavrakis, whom we work in close collaborations here in Fresnel, our collaborators in modeling these PSF engineering methods, which can be very powerful and which will continue to work hard, and our students in experimental developments. And I thank you for your attention.